Today, the point of my discussion is whether the entire genomics community who have been successful in decoding genome, especially the human genome, and then they deciphered the human genomics into letters like A, C, T, G. These letters, 4 letters, are actually the names of the proteins in our code, and these letters are sequenced like that. So protein sequencing in a linear way, and then terming it as a genetic code is actually genomics. And these letters are, proteins are sequenced in such a way which defines our genome. I have read the recent book, The Song of Cells, rather The Song of This Cell by Dr. Siddharth Mukherjee. This book has created a lot of questions in my mind, and I would like to discuss those things today. The first thing is these sequencing of genomes. When the CRISPR technology is saying that genes can be edited, it means these letters can be removed or replaced with a new letter in terms of genomics code. So my point was that this is a great breakthrough in the genomics, but I have one thing to tell, that with this kind of scientific tools available to genetics scientists of genomics, which they learned from one bacteria in the gut, which used to edit the genes of the virus to learn how to save from the viral attacks. So this technology of a bacteria was actually learnt, and then it was copied to the genomics, and that is how the Nobel Prize was also given to, and this is called CRISPR, where the genes can be edited. It means the letters in billion of letters sequenced in a linear way, and then A, C, T, G, G, C, T, A, T, whatever is there, formation. So what genetic scientists are considering is that the way these letters are proteins, in fact, are sequenced one after other, have keys of human life. They decide how we age, they decide how we will be vulnerable to particular disease, and in that process they make us vulnerable as well as safe from many things. And in fact, the key of body lies in this genomics. So after reading the book of Dr. Siddharth Mukherjee, I could infer certain things which I would like to share with the scientific community, because what I perceive is that these letters, maybe ACTG, whatsoever is that. So don't read them as letters. These letters must be constituting some words. Means block of proteins may be constituting another block of proteins if they are akin to another block. So what I would like to advise the scientific community, if they have not tried to do these things, first thing is that these letters can be replaced. Then these letters should be read in terms of words, means block of proteins, and then block of blocks. So in a simple word, a letter constitutes a word constitutes a sentence, and sentence constitutes a story constitutes a book. The book of life in genetics should be interpreted like this. So genomics, gene scientists should try to analogize this metaphor, that how letters in the words, into sentences, into stories, into book of life should be read when they are trying to understand the genetic code and that based dynamics of the body. So this is one such thing which emerged in my mind. Second thing is that there is still a lot of doubt and lot of mystery exists whether the genetic code is sanctum. Sanctorium means it cannot be altered naturally. Whether it gets impacted by the environment, this is yet to be learned. So, what I would like to put my mind on this is that let the scientist should create a model genome. For example, there are a lot more civilizations, places where there are centenarians are there who have no bigger disease in their life. Their genomics, their gene code should be modeled, and a kind of model genomics should be prepared. And with that, we can compare the genomics of normal human being, what are the vulnerabilities and what are differences. And these differences, if we have the sample of this centenarian's gene, maybe 100, and then we compare the people who have been facing chronic disease or acute disease or several things, and then we can easily understand what went wrong with their genomics, and what makes us aging, how what triggers the aging, which genomic code actually uh, uh, gives bad communication for cell replication and cell multiplication, cell duplicator. All these things can be easily understood if we have a model genome in terms of health and age. And this can be further because it's very difficult to experiment the human being. So I would like to especially the anti-aging and other things of genomics, we can try to understand it from understanding the gene code of say those creatures or insects or flies which have a lifespan of maybe 10-20 days. So if we understand what is the anti-aging gene in that, from there we can get the, because genetically there is hardly any variation in different species. So proteins of these flies, I mean 10 to 15 days, if you are able to increase their age maybe for 1 week, for 2 weeks, it means we have doubled their life. So understanding that protein and then trying to understand whether that protein which caused their life increase, whether has been existing in the human genome, if that is existing, and then engineering that to create a kind of better gene, better genomic code for longevity and all that. So that can be another approach. See, I am not a scientist. I am a basically humanity student, and when I am reading these great people like Siddharth Mukherjee or other cell scientists, the idea comes in my mind that what happens, I'm not criticizing that, what happens, scientists being very, very specific in their approach, sometimes somebody from outside, from humanity side, from philosophy side, brings certain things which is totally a new perspective, new context to their learning, and then if it is useful in terms of promoting some consequences and helping the scientists to discover a new horizon of knowledge, I think that is the purpose which I wanted to share with this. Because what I mean to that, this sequence of protein ACTG is having lot of block dynamics. So these 4 letters are a block. Inside there is a sequence, and then the, I mean, inter or intra block sequencing, and maybe after certain gap, if that a letter comes in a block, it has something to do. Another thing is that how to understand whether the genomics which was fixed at the time of the birth, does it change with the lifestyle while interacting with the environment. This is this has not been actually replied by the scientist, and this needs to be studied. To best of my understanding, what happens, the human sensory organs and human mind in general decides a course of action of the life. For example, I can decide what to eat, what not to eat. I can decide whether I will be at particular place which has different issues. So these all things actually get into us in terms of hormonal impacts in us, and hormones are basically triggered by the perceptions at times because of our thinking, our thought processes, and then we create a hormone which actually goes and which must be giving some trigger to the genomics to create a particular sequence, or to delete certain sequence, or to blur certain communication of the cells. I think the hormonal dynamics on the cells will be one important impactor on cellular dynamics. Other will be the biological factors. For example, polar bear. For example, those who live in cold areas, in million, billion years, their color changes. Those who dark, their color is different. So this kind of cellular changes in genetic code must be directly adopted by the body and transferred to the genetic code, and to some extent that is quite there. So, but this takes maybe centuries or million of years to actually translate that geography into genetic code, and then once it has been inducted into genetic code, then genetic code multiplies it irrespective of the biological environmental factors. So that is what I infer out of that. So hormone thought process must be having some transactional impact on our genetic codes. So this needs to be studied by the cellular scientist, genome scientist, and that is what I wanted to deliver this lecture for that. Thank you very much for listening patiently. My thought process may not be exactly scientifically true, but I am giving just a context to understand, or to infer, or to devise mechanisms to understand the impacts of these, because this cellular knowledge, cellular science is actually key to our entire medical world. Future medicines will be actually cellular medicines. Already there, all medicines are actually cellular. Even when we take antibiotics, what it does, it interferes with the bacterial cells while not touching our main cells. So actually we kill the bacteria, and then so it operates at cellular level. So many such medicines which are actually not designed cellular level, but in a very piecemeal they have been designed and they are successful. Maybe vaccines, etc. These are all. And then there is a whole lot of immunotherapy, etc. We can learn the immortality from cancerous cells, why they are immortal, how they, if they get energy, they can multiply unlimited. And then I recently read that somebody actually, some scientist has converted these immortal cells against the cancerous cells. I mean, killing the cancer by the cancerous cell. So a lot more things are in the offing, and I really congratulate the Genome project makers and funders and doers. And I personally think that genetics should be considered to be the biggest scientific investment if any country want to excel. And country like India, which has huge stock of top scientific brains, should encourage cellular scientists, and government should increase a lot of funding to cellular researches, and then integrating these cellular researches with the medicine will be the great opportunity for us, because the way human lives are being challenged by the bacteria and viruses. Recently, we had a pandemic. These things will be coming. So, the more we have the knowledge of the cellular dynamics inside us, and maybe the crop, maybe the wildlife, maybe anything. Once we understand genetics, we can easily maneuver that for the betterment of the humanity and public interest. So my one appeal to all those moral scientists is that let us not be very much scared about the ethics, because knowledge needs to be upgraded, knowledge needs to be researched, and then how to use that will be later on. But discarding any scientific investment or scientific breakthrough at the beginning is actually blunting the knowledge growth. So I will always respect the ethical boundaries, but research should be kept away from ethical boundaries, because ultimately we will learn and then we will decide. We will invent the knife and then we will decide that yes, knife should not be used for cutting the throat. But even if we have not invented the knife, and then we, how we will devise the mechanism? So learning is a very ethics neutral application of the learning is actually ethics question, but learning per se should not be discouraged. Thank you very much for listening me patiently. Thank you. Have a nice day. Thanks.